Hello, I am Jason Valent. Thank you to the American Society of Hematology for allowing us to present on the safety, tolerability, and efficacy of CAEL-101 in AL amyloidosis patients treated on a phase 2 open-label dose selection study to evaluate the safety and tolerability of CAEL-101 in patients with AL amyloidosis. CAEL-101 is an investigational IgG1 monoclonal antibody that targets the misfolded light chains of AL amyloid fibrils. CAEL-101 specifically binds to a conformational epitope present on both human kappa and lambda light chain amyloid fibrils, but not normal light chains. Phase 1 study established the safety and tolerability of monotherapy CAEL-101 as a weekly treatment for 4 weeks, up to a maximum tolerated dose of 500 milligrams per meter squared in relapsed refractory patients. No dose-limiting toxicity or drug-related deaths were seen. Cardiac response was seen in 67%, and renal response was seen in 33% of evaluable patients. While novel combination anti-plasma cell therapies continue to emerge, there is a critical need for therapies that target amyloid fibril removal to prevent and reverse organ damage. The hope is that these therapies will lead to a reduction in early death and improvement in long-term survival. The study design of CAEL-101-203 is a 3+3 design with dose escalation from 500 milligrams per meter squared to 750 milligrams per meter squared, and then to 1,000 milligrams per meter squared, which was the goal dose in combination with Cyborg-D. IV infusions were over 2 hours with pre-medication with acetaminophen 1 gram and diphenhydramine 25 milligrams orally. CAEL-101 was dosed day 1, 8, 15, and 22 of the first 28-day cycle, then every other week for the remainder of the study. Primary outcome measures were safety and tolerability of CAEL-101 when given in combination with Cyborg-D, and established the recommended phase 3 dose of CAEL-101 in combination with Cyborg-D. Secondary outcome measures were to evaluate the serum pharmacokinetics of CAEL-101 when given in combination with Cyborg-D. These data are presented in abstract 2277. Given the results of the Andromeda study, Part B of this trial will evaluate CAEL-101 in combination with daratumumab and Cyborg-D. The dosing of Cyborg-D was as follows: bortezomib 1.3 milligrams per meter squared subcutaneous, cyclophosphamide 300 milligrams per meter squared with a dose cap at 500 milligrams IV, and dexamethasone 20 milligrams IV were given weekly, day 1, 8, 15 of a 38-day cycle for the first cycle to align treatments with the CAEL-101, and then weekly days 1, 8, and 15 of a 28-day cycle for up to a total of 6 cycles, provided hematologic response was adequate. CAEL-101 was continued every other week as long as patients were tolerating and deemed to be deriving benefit. The key inclusion and exclusion criteria were that newly diagnosed and relapsed disease were permitted. Patients meeting criteria for multiple myeloma and Mayo stage 3B patients were excluded. This highlights patient and disease characteristics. Patient ages were representative of typical AL amyloid population, with ages even as high as 80. There were more men than women. But even though Mayo stage 3B patients were excluded, patients with NT-proBNP levels as high as 18,696 and troponin T levels as high as 0.106 were treated without cardiac toxicity. The safety and dosing summary. CAEL-101 treatment duration was from 40 to 276 days. The data cut occurred on November 1st, 2020. CAEL-101 was shown to be safe and well tolerated in combination with CyBorD with 1,000 milligrams per meter squared, established as the recommended phase 3 dose for the CAELM CARES ongoing trials. No dose-limiting toxicity or treatment-related discontinuation occurred. 2 patients that were escalated to 1,000 milligrams per meter squared were dose reduced to 750 milligrams per meter squared. One was due to diarrhea and the other due to vomiting. The patient with diarrhea did not have amyloid identified on colonoscopy. The patient with vomiting has and had previously biopsy-proven amyloid in the stomach and duodenum. Symptoms in both patients resolved with dose reduction to 750 milligrams per meter squared. Treatment is ongoing, with 40% of the patients having at least 15 infusions of CAEL-101. Importantly, no late cardiac safety signal has been seen. No infusion reactions were seen, and the pre-medication was discussed earlier. Regarding treatment emergent adverse events. From this slide, I would highlight rash, diarrhea, and nausea as potential treatment emergent adverse events. None of these were higher than grade 2. What is interesting is one of the patients with rash had also noted hair growth in areas where he lost his body hair around the time of his amyloid diagnosis. Regarding organ responses, the kidney organ responses were robust in most patients, and all kidney patients met Palladini criteria for organ response. Most interesting is the ongoing 76% reduction in 24-hour urine protein in a patient who achieved a very brief hematologic partial response while on CyBorD. CyBorD was discontinued after 6 cycles in this patient, and within 1 month, the patient reverted back to a hematologic stable disease. Despite this and no additional anti-plasma cell therapy to date, deepening kidney organ response continues. Cardiac organ responses by NT-proBNP criteria were not as robust as seen in the phase 1 study of monotherapy CAEL-101. Perhaps longer duration of therapy or therapy not concurrent with corticosteroids will show improved cardiac organ responses over longer follow-up. As a result of the safety demonstrated in this study, the international phase 3 trials in Mayo stage 3A and 3B patients have opened, and I would encourage trial participation in these most important studies. In summary, CAEL-101 dosed at 1,000 milligrams per meter squared is the recommended dose in combination with CyBorD for the ongoing randomized double-blind phase 3 trials. Organ responses, particularly in the kidney, are common even in relapsed patients. Only 1 patient is no longer on study due to the need for change in anti-plasma cell therapy due to inadequate hematologic response. And most importantly, organ responses have been seen even without ongoing hematologic partial response. We would like to acknowledge the following. Obviously, the patients and their caregivers, Dr. Alan Solomon and Dr. Suzanne Lynch, the research and nursing staff in the plasma cell disease group at the Cleveland Clinic, the Cleveland Clinic COVID-19 protocols, funding from CAELM Biosciences, medical monitors Michael Kerman and Margaret Ziola, John Salowski and Mark Harnett from CAELM, and the co-principal investigators on this study listed here. With that, I'm happy to take questions.